Today we are going to discuss sterilization methods. So there are two types of sterilization methods, physical and chemical. Physical methods include heat, radiation, and filtration. The heat can be of two types, dry heat and moist heat. Radiation has ionizing radiation like X-rays, gamma rays, and cathode. Where, on the other hand, there are non-ionizing radiation like ultraviolet radiation. Filtration can be membrane filtration and glass filtration. The dry heat can be dry oven or incineration. Moist heat can be pressurized steam or boiling water heating pasteurization. The chemical methods include gas and liquid. Under gas comes formaldehyde and ethylene oxide, and under liquid comes ethanol, phenols, aldehydes, and halogens. Sunlight has an active germicide effect due to the presence of ultraviolet rays. It reduces the number of microorganisms in water, tanks, lakes, etc. Whereas dry heat induces the denaturation of protein, oxidative damage, and toxic effect due to the high levels of electrolytes. It can also damage DNA of the microorganism. Moist heat kills the microorganism by denaturation and coagulation of proteins. Methods of dry heat sterilization. Dry heat provides long duration and reduced temperature. This is how red heat method is used. The inoculation loop, wires, forceps tips, needles, they are needed to be sterilized to inhibit microbial contamination. These instruments are held in the flame of a Bunsen burner until they become red hot. Flaming. Glass slides, scalpels, and mouth of culture tubes or conical flasks are passed through the Bunsen flame without allowing them to become red hot. Incineration. This procedure is used to reduce the infective material into ashes by burning. Hot air oven. 160 degrees for 2 hours is used for sterilization, 170 degrees for 1 hour, 180 degrees for 30 minutes. For example, sterilization of glassware, surgical instruments, chemicals such as liquid, paraffin, etc. Moist heat sterilization is under 100 degrees. First is pasteurization. Sterilization of milk. It works for all non-sporing pathogens. Second is boiling. This method can kill vegetative cells and boiling for about 10 to 30 minutes. For temperatures above 100 degrees Celsius, autoclave is used. This is the autoclave instrument. The steam above 100 degrees has better killing capacity than dry heat. When steam comes into contact with the cooler surface, it condenses into water and releases heat. It's like a modified pressure cooker. At the time of sterilization, the cylinder is filled with water and it is kept for some time for preheating. The temperature will increase eventually along with pressure, and when the temperature reaches 121 degrees, filtration is a process used for sterilizing those materials which are unable to withstand heat. And there are several types like candle filter used for purification of water, membrane filters which are made up of cellulose esters and are used for water analysis, 0.015 to 12 micron. The 0.22 micron filter is most commonly used. Radiation, ionizing radiation and non-ionizing radiation. Ionizing radiation is called X-ray and cosmic rays, and non-ionizing radiations are UV rays and infrared radiation. Ionizing. Due to high penetrating power, these radiations are lethal for cells. The bacterial cells are killed by damage in the DNA. Non-ionization. UV radiation of about 240 to 280 nanometers, bactericidal capacity, and the UV radiation causes protein denaturation and interferes with DNA replication of bacteria. Sterilization of operation theaters, laminar airflow, etc. is done with the help of UV radiation. Chemical method. Antiseptic and disinfectants. The properties of a chemical, antiseptic, or disinfectant are following. First, high penetration power. Second, it needs to be chemically stable under both acidic and basic environments. Alcohol, which is R group followed by an OH group. Ethyl alcohol and isopropyl alcohol are frequently used as chemical agents for disinfection. 70% ethyl alcohol is the standard concentration which is used for disinfection. Aldehyde. It can be used in both aqueous and gaseous form. A 10% formalin solution is a standard chemical disinfectant And sterilization of bacterial vaccines. It is used for prevention of tissues for histological examination. Phenols. They can kill microorganisms by cell membrane damage, for example, Lysol, disinfecting glasswares and floors. Halogens, like chlorine and iodine, they are commonly used as disinfectants. Chlorine is used in water supplies, swimming pools, and iodine is used as a skin disinfectant. Oxidizing agent, hydrogen peroxide. It is effective against most organisms in the concentration of 3 to 6%. However, it kills spores at higher concentration, that is 10 to 25%. That's all for the video. Thank you.